All right, chapter 15. So now is when we're going to start getting into our metabolic processes, and we're starting with glucose catabolism, which, as we just mentioned, is called glycolysis, breaking down glucose. So, there we go. So just as a general note before we get too far into this, for all the upcoming chapters, they tend to be a lot of pathways that in some ways you just kind of have to memorize certain parts of them. So as like a review, like with us together as a course, the review material is going to be shorter, let's say, because it's sort of more on you all to actually remember all the bits and pieces. Of course, we will have reviews for all the material, but the reviews will be shorter, and it's a little bit more memorization, which if you like memorization, that's great. If you don't like memorization, it might be a lot. But my goal here is to help you sort of understand why all this stuff is happening so that it's less just straight memorizing and sort of understanding how and why, which can help you along the way as you're trying to put all the pieces together in your mind. So, that being said, let's start with the overall reaction for glycolysis. So if you're looking at this and thinking, wow, I don't know, that's a lot, what is happening in glycolysis? You can sort of start with what you know. So we just talked about that's breaking down glucose, right? So we start with glucose on the left side, it has to be the beginning. Glucose has 6 carbons. What are we breaking down the carbons in glucose into during glycolysis? Yeah.
At the end of glycolysis?
Yeah.
2 pyruvate,
yeah.
2 ATP, and 2 NADH?
Yeah, overachiever, you got all of it. Yeah. So glucose itself is just we're talking about the carbons here. So that's the 2 pyruvates, right?
Oh.
So glucose has 6 carbons. We're breaking it down into 2 pyruvates. Each pyruvate has 3 carbons. So we're basically breaking each glucose molecule in half. And then you also talked about the energy we're making, right? You said we make how many ATP? 2 ATP. So that's on the product side. What would be on the reactant side for those ATPs? Yeah, ADP plus PI. So that's the energy we're getting out of it. And then the other thing that's involved, the other main thing that's involved are our electron carriers. So which electron carrier do we have and how many? Yeah, exactly. I believe it's just 1 actually, but yeah. So we have NADH as the product and NAD plus as the reactant. So those are the 3 main items I want you to think about for all of the reactions we're going to talk about. Where are the carbons going? What is happening in terms of energy synthesis or breakdown? What is going on in terms of electron carriers? A lot of the reactions also have, water and protons, stuff like that. I'm less concerned about you memorizing. They're there to help balance the equation, but knowing these 3 parts for all of the metabolic reactions we're going to talk about are going to help you understand what's kind of going on. Yeah. I'm pretty sure, is it not? I can go check. Yeah. Does it say 2?
Yeah.
Okay, maybe I forgot. My bad. It's 2. Any other questions? Okay. So let's start talking about the enzymes in glycolysis. So we know there are 10 steps in glycolysis. We break them down into 5 steps each, stage 1 and stage 2. So on this slide we have the stage 1 enzymes. On the next slide we have the stage 2 enzymes. And we talked a few chapters ago about the 6 different classes of enzymes: oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. So I would like you all to take several minutes. You can look back in the slides to look at what reaction each enzyme is catalyzing to help give you ideas for what's happening, and figure out what type of enzyme each of all of our glycolysis enzymes are, what kind of enzyme each one is. So everybody take a few minutes and look through it, and then we'll go over it together. Alright, so let's start from the beginning with hexokinase. So what kind of an enzyme is hexokinase? Yeah.
It's a transferase because it's a kinase.
Yeah, exactly. So hexokinase. Go ahead.
Also transferase.
Yeah, so great point. So all the kinases are transferases. They're taking a phosphate from an ATP molecule and transferring it onto the substrate. So that's what a kinase does. All kinases, like you said, are transferases. So right away we can look for anything that says kinase, and we know that's a transferase. And even if we go on to stage 2, we have more kinases, right? We have phosphoglycerate kinase, we have pyruvate kinase. All the kinases are transferases. So excellent start. So let's go back to step 2, phosphoglucose isomerase. Yeah. Yeah, so of those 6, oh right, that's an isomerase, sorry. Yeah, so it's an isomerase, right in the name, right? So very helpful. Which other one on this list is also an isomerase? Yeah. So aldolase is actually, yes, 5, triose phosphate isomerase. So again, it's right in the name. Isomerase is an isomerase, very straightforward. What is another really common name that isomerases sometimes have that is in this list? Yeah. Like the enzyme's name. Yeah. Yeah, mutase, exactly. So phosphoglycerate mutase, that's another isomerase. So those ones are easy to identify what they are right from the name. So a mutase is another, so isomerases tend to have names like isomerase or mutase. And these are just rearranging the bonds of the atoms within a molecule. We're keeping the actual atoms the same. We're just moving them around within the same structure. All right, so we're almost done with the stage 1. We're just left with aldolase. So does anybody know what kind of enzyme aldolase is? Yeah. Yeah, it's a lyase. So what makes it a lyase? Yeah, exactly. So let's look at that one because it's a little bit harder to visualize than some of the other ones. Alright, so we have aldolase. So in aldolase, we're breaking a bond, but in the process of breaking that bond, we're forming a new double bond. And that's the definition of what a lyase does. So in this case, we're breaking this bond between carbon 3 and 4. We are, and then we're making a new double bond out of this oxygen. It becomes a carbonyl. So that's what lyases do. If we were breaking this bond but using water as a reactant and not creating a new double bond, that would be hydrolysis, a hydrolase, but that's not what we're doing here. So it's a lyase. All right. Okay, cool. So that's stage 1. So let's finish up stage 2. So number 6, glyceraldehyde 3-phosphate dehydrogenase. What kind of enzyme is that one? Yeah. An oxidoreductase. Yeah, how do we know that? Yeah, exactly. So the clue here, again, right in the name. So dehydrogenase, another really common name for oxidoreductases. All right, so that brings us to enolase. Yeah, it's also a lyase. Yeah, how do we know that? Yeah, exactly. So we have the same idea. We're breaking bonds, and in the process of breaking the bond, we're creating a double bond. So in between carbons 2 and 3, we're getting a double bond. And we're breaking these bonds to hydrogen and the hydroxyl group, creating a water. And I, again, want you to be really careful with this one, because if you think about the reverse of it, you might think it's a hydrolase, right, if you're adding a water. But remember, hydrolases add water to break a bond. So like one substrate becomes 2 products. And that's not what we have here. We have one, so like in the reverse direction, when we add the water in, we are not breaking the substrate into 2 pieces. We're just breaking that double bond. So it's a dehydration reaction using a lyase. This enzyme is not a hydrolase. Any questions about that? Okay. Sorry for this. Okay, so yeah, so that's all 10. And let me think. Yeah, so I guess the other thing to mention here is like a lot of these enzymes have very long names that you might not want to spell. You are welcome to use abbreviations as long as it's clear which one you mean. So like you could call hexokinase HK, right? That's the only one that you could abbreviate HK. But don't call like phosphoglycerate kinase PK, because pyruvate kinase is also PK. So feel free to use abbreviations, but be careful. Yeah, any questions about the classes of those enzymes? All right. So, of those 10 enzymes that we just talked about, which one is the most highly regulated? Yeah. Phosphofructokinase. Yeah, step number 3. Which reaction does PFK, so this one you can call it PFK, feel free, it's a very long word. Which reaction does it catalyze? Yeah, so what is it adding a phosphate to? Yeah. Did you want to add anything? Yeah, here's F6P. Yeah. Yeah, so we have F6P, that's fructose 6-phosphate. This kinase it's a transferase. It's transferring that phosphate from an ATP, like all kinases. And F6P, fructose 6-phosphate, we're adding a phosphate to the 1 carbon. So it becomes fructose 1,6 bisphosphate. So F16BP. And one of the phosphate groups is lost from the ADP, so we're left with an ADP. And yeah, and we're going to talk a lot about 2 different types of fructose bisphosphates, fructose with 2 phosphate groups. So this is the one that's involved in the actual glycolytic pathway. You can also call this molecule FBP1. So fructose, 2 phosphates, one of them is on the 1 carbon. you can assume the other one's on the 6 carbon, because for both of the ones we're going to talk about, the other one's on the 6 carbon. And so for this reason, you can also call this enzyme phosphofructokinase PFK1, because it catalyzes adding that phosphate to the 1 carbon. We're going to also talk about another enzyme called PFK2 that adds a phosphate to the 2 carbon. So start getting those together in your minds, I guess, now, before we all confuse ourselves. So this particular reaction, because it's very important for regulation, I would like you to know this. So for the other reactions, I want you to know the enzymes that are being used in the order, be able to sort of match them up with each other, but don't necessarily sort of memorize the full reaction for each step, aside from this one, if that helps anyone in terms of studying. Yeah. Yeah. Yeah, so it's just, think of it like a nickname. Let me go to the structure. Sorry, this is, close your eyes, everyone. Okay, so we start with F6P. This is fructose 6-phosphate. It has a phosphate group on the 6 carbon. PFK1, is adding a phosphate group to the 1 carbon. So we already know that we have a phosphate group on the 6 carbon, so we can call it FPP1 for short, because like this one is sort of assumed, if that helps. Yeah. And then we're going to talk, like I said, later on about FPP2 and PFK2 that are involved in regulation, which is also part of this chapter. So yeah. Close your eyes. Sorry. Okay. All right. Any questions about this before we move on to more about regulation? Okay. So now we have some allosteric effectors and other components that might help to increase or decrease the rate of glycolysis. So so I'd like everybody to take a few minutes and think about, for each of these, based on what we know so far, you should be able to reason through whether each one of these would speed up or slow down breaking down of glucose. So take a few minutes and think about it, and then we'll go over it together. Alright, so let's start with glucagon. So what is glucagon signaling telling us, and what does that mean in terms of whether or not glycolysis is going to happen? Yeah. Yeah, why would that be? Yeah, exactly. So glucagon is a signal that we have low blood glucose, right? So yeah, so first of all, if we want to do glycolysis, our reactant is glucose. If we don't have a lot of glucose, very difficult to do glycolysis, right? No starting materials, not going to go forward. So that is a really big reason why glucagon will help decrease the rate of glycolysis. And in particular, in your liver, when you get glucagon signaling, because that means you have low blood sugar, your liver is going to want to send out any glucose that it has to the rest of your body. So it's not a time to break down the glucose, it's a time to send it where it's needed. Alright, so how about insulin? Yeah. Yeah. So insulin is a sign of high blood glucose. So this is a time when we're going to be importing glucose into our cells. For either for storage or for breakdown, which in our case is glycolysis. Alright, how about ATP? Yeah. Yeah, why would that be?
I think it inhibits PFK.
Yeah.
Actually, like it uses PFK. PFK uses ATP, but like slows it down.
It does, yeah. So, so all of these, that's a really good point. So all of these different effectors are all acting on the activity of PFK, right? Because PFK is the main enzyme that's getting regulated during glycolysis. So that was a really good point. Thank you. Also, ATP, if you have a lot of ATP available in your cell, what is the point of glycolysis? Glycolysis is helping us make more ATP. And also help to make reduced electron carriers that are going to get sent through to ultimately help make more ATP during oxidative phosphorylation. So overall, glycolysis is a process for making more ATP when we need it. If we already have a lot of ATP, why would we want to make more? It's a waste of energy and work. So ATP is a sign of having high energy levels. So no need to make more. So we're going to decrease glycolysis. So how about ADP? Yeah.
Both ADP and AMP have opposite effects. So in this case, they lower energy and therefore increase.
Yeah, exactly. So this is a sign of low energy levels. or need for more ATP, so we'll increase the rate of glycolysis. All right, and how about F26BP? Yeah.
Would it decrease the
So it actually does that. That's okay. So that is one of the allosteric effectors of PFK1, our regulated enzyme. But F26BP, this one is the molecule that's also called FBP2. So, and this one is an allosteric activator of PFK1. And we'll go more into detail about that in, I think it's the next chapter, chapter 16. But for now, as a guess, like a logical next step, which hormone would result in the production of F26BP or FBP2, glucagon or insulin? Insulin, yeah. So activating PFK1, we're going to increase the rate of glycolysis, which we know also is the result of insulin signaling, also increases the rate of glycolysis. So you make more FBP2 as a result of insulin signaling. All right. Any questions about our activators and inhibitors of glycolysis? I just put pluses and minuses in case you just want to like look at it more quickly. All right. Any questions about anything we talked about so far? Or we're just thinking? Okay. So this is a blank glycolytic pathway. This is from one of your worksheets with the practice questions. We don't need to go over it together. This is one of those things where you had to take the time on your own to like remember each enzyme and the order that they go in. But it's helpful to sort of think of the pathway as a whole, or to think of each enzyme in terms of what reaction it's catalyzing. Because like we were talking about earlier, a lot of the enzymes, their names give you a clue for what they're doing. And that can, if you have information about how the structures of the molecules are changing, you can take an educated guess about what type of enzyme might be doing it, and that kind of a thing. So like this slide kind of goes together with the next slide. So the next slide has a list of all the enzymes and metabolites that are involved. And then you can, on your own, you can use this sort of word bank to fill in the blank pathway. And I think this is a really helpful way to study. And again, I just want to emphasize trying to use a logical understanding as much as you can, rather than just trying to memorize this, because this is a lot of stuff to just straightforwardly memorize, and we're going to be going over some other pathways in the next few chapters. So it will get overwhelming if you just try to memorize it without sort of understanding what's happening. So, that being said, I guess start now. But I'm not gonna, sit here while you do this. But I do recommend that as you study, that you work on sort of being able to fill in the blank pathway. And okay, so does anyone have any more questions about glycolysis? Yeah.
And you said, do you have to be memorizing the structures?
No. The one that I want you to be most familiar with is the reaction that PFK1 catalyzes, step 3. In terms of, I'm not going to make you draw fructose, but be able to, know where the phosphates are getting put and that kind of thing. Yeah, know the order of the enzymes and each of the metabolites that the enzyme is going to and from, substrates and products. As well as, very important to know which steps are going to give you reduced electron carriers, which steps result in generating ATP. There are 3 irreversible steps. It's helpful to know that those are irreversible because when we talk about gluconeogenesis, which is the opposite of glycolysis, a lot of the reversible steps are the same, but the irreversible steps obviously are different. So it's helpful to be able to know, okay, these are reversible. I know them for glycolysis, and they're the same for gluconeogenesis. So it's, you can already start to see step 1, 3, and 10 are your irreversible ones. So that's helpful. Maybe just start organizing it in your mind, that kind of thing. So, okay, so I wanted to, since we have some time, talk about having a quiz 12. So we have 11 quizzes so far. So the way the course works is you get to drop your lowest quiz grade. So if you'd like, we can just leave it at 11 quizzes. You won't be quizzed on anything past whatever chapter that was, 12. That was on quiz 11. Chapter 13. Yeah, I don't know the numbers anymore. Or if you want extra practice, we can have a quiz 12. That will be on chapters 15 and 16, as in what we discussed today with glycolysis and what we'll talk about on Thursday. If you do want to have that 12th quiz, then your quiz grade will be out of your highest 11, and I'll drop the 12th. So, I guess I'm seeing nodding. So does that mean we want a 12th quiz? Okay, so we will have a quiz 12. So that will be after we finish discussing chapter 16 on Thursday. That'll be due a week from Thursday, whatever date that is, like December 3rd or something. That's not what it is, but it's whatever date that is. Okay. Any other questions? All right, so I guess get ready to talk about chapter 16 on Thursday. And don't forget tonight your quiz 10 is due. And if you're submitting a rough draft, that's also due tonight.